Chapter 27. Tissue fluid and edema. Definition. Tissue fluid is the medium in which cells are bathed. It is otherwise known as interstitial fluid. It forms about 20% of extracellular fluid, ECF. Functions of tissue fluid. Because of the capillary membrane, there is no direct contact between blood and cells. And, tissue fluid acts as a medium for exchange of various substances between the cells and blood in the capillary loop. Oxygen and nutritive substances diffuse from the arterial end of capillary through the tissue fluid and reach the cells. Carbon dioxide and waste materials diffuse from the cells into the venous end of capillary through this fluid. Formation of tissue fluid. Formation of tissue fluid involves two processes: filtration, reabsorption. Filtration. Tissue fluid is formed by the process of filtration. Normally, the blood pressure, also called hydrostatic pressure, in arterial end of the capillary is about 30 mm Hg. This hydrostatic pressure is the driving force for filtration of water and other substances from blood into tissue spaces. Along the course of the capillary, the pressure falls gradually and it is about 15 mm Hg at the venous end. Capillary membrane is not permeable to the large molecules, particularly the plasma proteins. So, these proteins remain in the blood and exert a pressure called oncotic pressure or colloidal osmotic pressure. It is about 25 mm Hg. Osmotic pressure is constant throughout the circulatory system and it is an opposing force for the filtration of water and other materials from capillary blood into the tissue space. However, the hydrostatic pressure in the arterial end of the capillary, 30 mm Hg, is greater than the osmotic pressure. And, the net filtration pressure of 5 mm Hg is responsible for continuous filtration, Fig 27.1. Starling Hypothesis. Determination of net filtration pressure is based on Starling Hypothesis. Starling Hypothesis states that the net filtration through capillary membrane is proportional to the hydrostatic pressure difference across the membrane minus the oncotic pressure difference. These pressures are called Starling forces, refer Chapter 52 for more details. Reabsorption. Fluid filtered at the arterial end of capillaries is reabsorbed back into the blood at the venous end of capillaries. Here also, the pressure gradient plays an important role. At the venous end of capillaries, the hydrostatic pressure is less, 15 mm Hg, and the oncotic pressure is more, 25 mm Hg. Due to the pressure gradient of 10 mm Hg, the fluid is reabsorbed along with waste materials from the tissue fluid into the capillaries. About 10% of filtered fluid enters the lymphatic vessels. Thus, the process of filtration at the arterial end of the capillaries helps in the formation of tissue fluids and the process of reabsorption at the venous end helps to maintain the volume of tissue fluid. Applied physiology, edema. Definition. Edema is defined as the swelling caused by excessive accumulation of fluid in the tissues. It may be generalized or local. Edema that involves the entire body is called generalized edema. Local edema is the one that occurs in specific areas of the body such as abdomen, lungs, and extremities like feet, ankles and legs. Accumulation of fluid may be inside or outside the cell. Types of edema. Edema is classified into two types, depending upon the body fluid compartment where accumulation of excess fluid occurs: intracellular edema, extracellular edema, intracellular edema. Intracellular edema is the accumulation of fluid inside the cell. It occurs because of three reasons: malnutrition, poor metabolism, inflammation of the tissues. Edema due to malnutrition. Malnutrition occurs because of poor intake of food or poor circulatory system, through which the nutritive substances are supplied. Due to the lack of nutrition, the ionic pumps of the cell membrane are depressed, leading to poor exchange of ions. Especially, the sodium ions leaking into the cells cannot be pumped out. Excess sodium inside the cells causes endosmosis, resulting in intracellular edema. Edema due to poor metabolism. Poor metabolism is caused by poor blood supply. Poor blood supply leads to lack of oxygen. It results in poor function of cell membrane and edema, as explained above. Edema due to inflammation of tissues. During inflammation of the tissues, usually the permeability of cell membrane increases. This causes the movement of many ions, including sodium into the cells, resulting in endosmosis and intracellular edema. Extracellular edema. Extracellular edema is defined as the accumulation of fluid outside the cell. Causes for extracellular edema. Abnormal leakage of fluid from capillaries into interstitial space. Obstruction of lymphatic vessels that prevents fluid return from interstitium to blood. Conditions which lead to extracellular edema: heart failure. Renal disease. Decreased amount of plasma proteins. Lymphatic obstruction. Increased endothelial permeability. Edema due to heart failure. Edema occurs in heart failure because of various reasons, such as failure of heart to pump blood. Failure of the heart to pump blood from veins to arteries increases venous pressure and capillary pressure. This leads to increased capillary permeability and leakage of fluid from blood into interstitial fluid, causing extracellular edema. Fall in blood pressure during heart failure. It decreases the glomerular filtration rate in the kidneys, resulting in sodium and water retention. So, the volume of blood and body fluid increases. This in turn increases the capillary hydrostatic pressure. These two factors together increase the accumulation of fluid, causing extracellular edema. Low blood supply to kidneys during heart failure. It increases renin secretion, which in turn increases aldosterone secretion. Aldosterone increases the reabsorption of sodium and water from renal tubules into ECF, resulting in the development of extracellular edema. Pulmonary edema. Pulmonary edema is the accumulation of fluid in pulmonary interstitium. In left heart failure, the blood is easily pumped into pulmonary circulation by right ventricle. However, the blood cannot return from lungs to left side of the heart because of weakness of this side of the heart. This increases pulmonary vascular pressure, leading to leakage of fluid from capillaries into pulmonary interstitium. It causes pulmonary edema, which can be life-threatening. Edema due to renal diseases. Generalized edema. In renal disease, the kidneys fail to excrete water and electrolytes, particularly sodium, leading to retention of water and electrolytes. So, the fluid leaks from blood into interstitial space, causing extracellular edema. Initially, the edema develops in the legs, but later it progresses to the entire body. Generalized edema. Edema due to decreased amount of plasma proteins. When the amount of plasma proteins decreases, the colloidal osmotic pressure decreases. Because of this, the permeability of the capillary increases, resulting in increased capillary filtration. So, more amount of water leaks out of the capillary. It accumulates in the tissue spaces, resulting in extracellular edema. Amount of plasma proteins decreases during the conditions like malnutrition, liver diseases, renal diseases, burns, and inflammation. Edema due to lymphatic obstruction. Lymphedemia. Lymphedemia is the edema caused by lymphatic obstruction. It is common in filariasis. During this disease, the parasitic worms live in the lymphatics and obstruct the drainage of lymph. Accumulation of lymph along with cellular reactions leads to swelling that is very prominent in legs and scrotum. Repeated obstruction of lymphatic drainage in these regions results in fibrosis and development of elephantiasis. Elephantiasis is a disorder of lymphatic system, characterized by thickening of skin and extreme enlargement of the affected area, most commonly limbs, legs, genitals, certain areas of trunk and parts of head. Edema due to increased endothelial permeability. The permeability of the capillary endothelium increases in conditions like burns, inflammation, trauma, allergic reactions, and immunologic reactions, which lead to oozing out of fluid. This fluid accumulates, leading to development of edema. Pitting and non-pitting edema. Interstitial fluid is present in the form of a gel that is almost like a semi-solid substance. It is because the interstitial fluid is not present as fluid but is bound in a proteoglycan meshwork. It does not allow any free space for the fluid movement except for a diameter of about a few hundredths of a micron. Normal volume of interstitial fluid is 12 L and it exerts a negative pressure of about 3 mm Hg. It applies a slight suction effect and holds the tissues together. However, in abnormal conditions, where the interstitial fluid volume increases enormously, the pressure becomes positive. Most of the fluid becomes free fluid that is not bound to proteoglycan meshwork. It flows freely through tissue spaces, producing a swelling called edema. This type of edema is known as pitting edema because, when this area is pressed with the finger, displacement of fluid occurs producing a depression or pit. When the finger is removed, the pit remains for few seconds, sometimes as long as 1 minute, till the fluid flows back into that area. Edema also develops due to swelling of the cells or clotting of interstitial fluid in the presence of fibrinogen. This is called non-pitting edema because, it is hard and a pit is not formed by pressing.